A-level biology 9700 paper 4 solved. This is the March 2016 paper 4 2, and some tips and helps in solving the paper 4, which is just a few days away. As we all know, the paper is for 2 hours, but this time is still very short for a paper 4 because you need to understand the questions, and the main thing is that the majority of the answers are in the question, but you need to read the question carefully and look for the answers. One, the rate of photosynthesis is affected by a number of environmental factors. Now, very simple to remember: TLC, temperature, light intensity, and CO2 concentration. So TLC is a very easy way to remember it. Now, you always look what is on the y-axis. Sorry, what is on the x-axis here? The x-axis is light intensity, y-axis is always the rate of photosynthesis. Now, when you look at this point A to this point here, whatever is on the x-axis, this is the limiting factor. So the first clue that I give you is that always look at the x-axis, and that is the limiting factor. So this red line is the limiting factor, and after that we say this light intensity is no longer limiting. Why? Because you see, if you put figures here 1, 2, 3, 4, 5, 6, 7, 8. So up to this point, light was the limiting factor. After this, even we are increasing the light, but the rate remains constant. So light is no longer limiting. So only from this point, like if I say this is point A and this is point B here, so this point A to B is the limiting factor, and this whatever is on the x-axis is the limiting factor. Now, let's look at the questions. The questions are: state the limiting factor in region A on the graph. Now, region A on the graph is naturally the light intensity. I told you whatever is on the x-axis, that is the limiting factor. Then explain what is meant by the limiting factor. Now, limiting factor definition, I've never been able to really learn it, and I find it difficult as well. When a process is affected by more than one factor, the factor that is in short supply and prevents further increase in the rate of the process. Like there's a factor, three factors, which I just told you: temperature, light intensity, and carbon dioxide concentration. So all these three factors limit the process. So when a process is affected by more than one factor, now if there's less light intensity, then temperature might be fine, carbon dioxide may be fine, but then that is the limiting factor. If temperature and light are enough, but carbon dioxide is in short supply, then we say carbon dioxide is a limiting factor. Then, of course, we come on to the part 3 of the question. So, explain why there is no further increase in the rate of the photosynthesis beyond C. So, after C, this point, why is there not an increase in light, and why is there not an increase in the rate of photosynthesis? You see, the rate of photosynthesis is remaining constant. That's why the line has become straight. So why is there not, that is what we have to understand, is that some other factor becomes limiting. And even if you wrote that, some other factor becomes limited, you actually got a mark for that. And that could be the other two factors, as I just told you, TLC. So if light is not the limiting factor, some other factor is limiting. Now that can be either CO2 concentration or temperature. Then the B part of the question says, for many plants living in temperate regions, the optimum temperature for photosynthesis is approximately 25 degrees Celsius. Suggest reasons why the rate of photosynthesis decreases at temperatures above 25 degrees Celsius. So rate decreases. Now read the question carefully. So Rubisco naturally temperature rises. The enzyme which is most important is Rubisco, that's denatured. Then we need enzymes for photolysis. So less photolysis, less ATP produced, less Calvin cycle, less carbon dioxide fixed. Why? Because we need the enzyme Rubisco for that. Then increase in transpiration, because naturally temperature is high, so more evaporation, more diffusion of water vapors out of the leaf. And then we could start off with photorespiration, in which Rubisco decides to combine RUBP with the oxygen, and the oxygen starts to be used. Respiration means the oxidation, oxygen is being used. So then, of course, the RUBP will not combine with carbon dioxide, so Calvin cycle will not occur. So stomata close, yes, in response to high temperatures to prevent the loss of water vapors, and the plant can start to wilt. And of course, when the stomata are closed, then reduce CO2 uptake. Naturally, if the stomata are closed, then that means they will be the carbon dioxide will not diffuse into the leaf. The IUCN Red List provides information about the conservation status of species throughout the world, including the American badger and the black-footed ferret. So let's talk of the badger and the black-footed ferret. Whatever. Figure 2.1 shows an American badger. Here we have the American badger, Badger. Make it a point to write on the paper. And this is a ferret. They both look rather cute animals. Figure 2.3 shows the IUCN conservation status of the American badger and the ferret. So let's just say the badger, forget the American and the black-footed one. So badger and ferret, let's make it simple so that we can understand the question. 1987, 2013, American badger, conservation status, least concern, American badger, least concern, 2013. So 87 we also they were least concern, and then also their least concern. Now then after that is near threatened, vulnerable, endangered, near threatened, vulnerable, endangered, critically endangered. We have all these different things in it. But the ferret is now extinct in the wild. And the black-footed ferret here was endangered at this time. So we can see how it has changed over a period of time, that in 1987 it was least concern, and in 1997 the black-footed ferret was extinct in the wild, while in 2013 the badger is least concern, but the black-footed ferret is now endangered. So this was extinct in the wild, and this was, of course, now you have to read the question. American in 87 and in 2013. So we've got, this is the data for 87 and this is the data for 2013. In 87, the badger was least concern and the footed ferret was extinct. In 2013, the American badger is least concern, still the same, but the ferret is now endangered. Now everything is in the question. The badgers and the ferrets are both predators. Badgers feed on prairie dogs and a range of other animals. So they have two or three different foods on which they rely on. And the ferrets feed almost entirely on prairie dogs. So this is only one food that they eat. Like for instance, we can say I sometimes have a burger from McDonald's, sometimes I have it from KFC, sometimes I have it Hardee's. So you have a choice between the burgers in three different places. So badgers do not have any animal predators, so nothing eats them. Ferrets are preyed upon by the badgers and several other predators. So ferrets are eaten up by a large number of other animals. So read the question, understand it, do the story to yourself. Suggest reasons why ferrets are endangered but American badgers are not. So the question was, look, it's giving again the question. Ferrets feed on entirely on prairie dogs. This is the point. So answer marking key. Ferrets feed on one type of prey. Now, what should have happened to these prairie dogs? Black-footed ferrets feed on prairie dogs. So reduction in prairie dog population. So this decreases the number of ferrets when they have less food, so less of the ferrets. But the other ones, the ferrets, are preyed upon by this. So ferrets have many predators. Again, from the question we picked it up. So the ferrets are preyed upon by the American badgers and several other predators. So same thing we are rewriting here. In this point, ferrets have many predators. Predators decrease, ferrets more than the badgers. So the predators are eating more of the ferrets and less of the badgers, so that's why their numbers decrease and the badgers remain the same. In 1987, this is the B part of the question we are doing. In 1987, the world population of black ferrets consisted mainly of only 18 animals living in captivity. A number of different agencies worked together to prevent the extinction of this species. Their goal was to produce young black-footed ferrets to be released into the wild. The survival and breeding of the animals in the wild would then be monitored and supported. So this is now a form of captive breeding that you would goal was to produce young ferrets to be released into the wild. The collaborating agencies included local government, universities, zoos, and native tribes that own undeveloped reservation land. Now, outline how these different agencies could contribute to successful conservation of the black-footed ferret. Create protected zones, carry out research, run captive breeding programs because we had zoos here, so we can have that there. Then they could do research. Why? Because we had a university there, so we could do research. Then Native American tribes provided suitable habitat. They lived in whatever the forest or wherever they lived, they probably gave them the right habitat for them to survive. And then, of course, you could do IVF. IVF was one way by in vitro fertilization, monitor the wild populations, and then ensure that the numbers increase and we can put them back into the environment. Then it says black-footed ferrets were released at three different locations in the wild at different times. Each population was established from the captive bred animals. Captive breeding means you breed them in the zoo and you look after them and then you introduce them back into the wild. So they were released at three different locations at different times. Now, figure 2.4 shows the population sizes of black ferrets at the three release locations. So you look at the graph and you look at it very carefully and you write down the figures for it. So here we have the population size in number of individuals, and on the x-axis, what do we have? We have the year. So nothing very different or very weird. We just have the year, and you can see how. And what was the question is that they were released at three different locations, and each population was established from captive bred animals. Okay, so let's look at this now. Now, I give you the final thing. I say, okay, South Dakota, what were there? 270. I've written the points here. Wyoming, 80. Arizona, 30. And South Dakota, 270. So look at it carefully. Write down the points. See how the South Dakota has been increasing very, very, very sharply. Then we have Wyoming. Now let's look at Wyoming. That's constant. And then it goes like this. And then it goes like this. And then uh, we have the other one which is very nominal, it remains very low, and this is the Arizona story. So the three stories, we go through it, we look at it, we write down the figures for it in a number of places, but I like this at the end, or I usually do either in the beginning or in the middle. I could have done in 2000, I could have done in 2002. They were also good figures to take. Now I often tell you this is describe. Describe is no biology. So we don't want you to give us any biology. You just have to give us things of the graph and says South Dakota increased continuously, then Wyoming constant initially, then decreased to 10 before increasing. Arizona population very low, then recovering. And in 2004, Wyoming and Arizona, and I gave the figures of the population. So describe is no biology. You just give me points of the graph. You just look at the graph and you give me all the points. South Dakota increased. You can see how South Dakota increased. Then the second was Wyoming constant is the one in red. Wyoming constant. And then you can see initially the decrease to 10, like here you see the red line which I've drawn you, initially decreased to 10 before increasing. So you can see this. And then Arizona was, you look at the Arizona population, Arizona population very low, then recovering. So it was very low and then it's recovering slowly. So describe no biology. Table 2.1 shows information about the gene pool of the population of black-footed ferrets and the leg sizes of the black-footed. So gene pool and leg sizes at each release locations in 2004. All three populations were started by animals from the same captive population. So there's not a lot of gene mixing. So you can think of inbreeding and inbreeding depression. In this original captive population, 100% of the genes surveyed showed polymorphism. That is, they had more than one allele. And I gave a simple example of HbA and HbS. There are two alleles. The mean number of alleles per gene locus was 2. The mean number of alleles per gene locus was 2. So they want three alleles. Like you remember, we did in O levels, blood group, there are three alleles, IB, IA, and IO. But here it says that they had the mean number of alleles per gene locus was just 2. So there was either big T, small t, or there was big B, small b. So just 2. The population of South Dakota location in 2004 maintained the same level of genetic variation and the leg side data as the original captive population, but the populations in Wyoming and Arizona showed changes. So table 2.1 gene pool data, percentage of genes that are polymorphic is 100, 4300. Mean number of alleles 2.0, 1.30. This one and this one is the same. This is the same. Then leg size data, mean length was 69.4. So this one and this is the same. Then this and this is the same, and this is different, and this is different. So look at the table. Like I'm doing is mark them with a pencil or a pen. You can do that. Rough work is allowed on the paper. It doesn't matter what you wrote. We're going to read the answer and then figure out what you've written. Then part 2 of the question was use table 2.1 to describe how the gene pools and leg sizes of the Wyoming and Arizona Blackfoot ferret have changed compared to the original captive. So Wyoming, you can see here, Wyoming is here, bone length smaller. So look at the bone length. Lower bone leg was 68 and 56.7. Here we have these two figures. So Wyoming ferret bone length smaller. So this is something which you needed to figure out yourself, right? Then Wyoming has lost the genetic variability or different alleles. So it has probably lost that genetic variation. And Arizona gene pool has increased because you see, this is now nearly that of South Dakota. So Arizona gene pool has increased. Then, of course, the next part of the question was with reference to figure 2 suggest reasons for the changes. So that was just the main thing, and then now you have to give reasons. So reduced size may be due to less food available. So less food available, so less bone size, very small population, so smaller gene pool, and Arizona extra allele due to chance, random mutation, but they seem to get a little more and they're slightly different. Whenever you see something which is slightly different, I mean, always think of mutation as the marking scheme point. Then part D of the question. In 2008, some black ferrets were born in captivity as a result of IVF using frozen sperm that had been stored for several years. Explain the benefits of using frozen sperm in captive breeding programs. Increased number of potential mates, larger gene pool, sperm transported to other zoos, frozen sperm act as a gene bank, alleles available from animals no longer alive. So these are the general points which we give for all these like seed banks and frozen sperms and botanic gardens and all that. So this is something which is very general and we write this nearly in every mark scheme. Then question 3. Myofibrous and striated muscle consist of contractile units called sarcomeres. When an impulse stimulates striated muscles to contract, calcium ions are released from the sarcoplasmic reticulum. Describe how the release of calcium ions leads to the contraction of a sarcomere. So calcium released from sarcoplasmic reticulum binds to troponin. Troponin changes shape. Tropomyosin is displaced. Very good word. Tropomyosin is displaced. Myosin binding sites exposed. Myosin head now binds to actin. Myosin pulls the actin. So very simple mark scheme and very plausible and very answerable. So makes sense. The many-banded crate Bungarus multitinctus is a venomous snake. So whatever is a venomous snake and shows a many-banded crate. The venom forms the main branded crate. The venom from the many branded crate contains bungarotoxin. In animals, they are bitten by. Now, whatever these words are, even if you can't read them and it daunts you, just don't read them, just say toxin. The venom from the many crate contains toxin. In many animals that are bitten by the snake, the venom acts as the neuromuscular junction, causing muscle paralysis, loss of muscle action function. State how bungarotoxin may cause muscle paralysis. A very vague question, but makes sense because they talked of the neuromuscular junction. So, reduces the release of acetylcholine. What is ACH stands for? Acetylcholine. Now, you can write that as a standard abbreviation. Now, this is a neurotransmitter. So, reduces the release of acetylcholine, less binding of acetylcholine on the postsynaptic membrane, therefore, no depolarization, binds to receptor on sarcolemma, prevents ACH from binding, inhibits depolarization of sarcolemma, inhibits acetylcholinesterase, so of course that won't break down, ACH not broken down, permanent depolarization of the sarcolemma. So acetylcholine will not be broken down. Here we have acetyl. ACH is the abbreviation of acetylcholine. Question 4. The process of gametogenesis in male and female vertebrates, including humans, involves meiosis, reduction division. Describe how gametogenesis differs from human males and females. Now, I just made this table and I think that makes it so easy to explain it. Female begins before birth, begins at puberty, one ovum, four spermatids, interrupted process, continuous process, then greater number of gametes produced in males and can produce gametes to a greater age. Then B part of the question says the figure 4.1 shows a Komodo dragon. Whatever, this species of lizard is only found on 5 Indonesian islands. So this huge thing is found in the species of lizard is only found in, so it won't be that huge then I think. In 2006, two captive females, Komodo dragons, captive female A and B, in British zoos, each produced healthy offsprings 1 to 4, despite never having mated with a male. Female B was later mated with a male Komodo dragon C. So B mated with C and produced an offspring which was 5. Now, genetic analysis was performed on the two females A and B, their offsprings 1 and 4 that had no father, and the male C and the offspring 5 produced by the mating between B and C. The results of the genetic analysis are shown in table 4.1. Here we have table 4.1. Different alleles at 4 gene loci P, Q, R, and S could be distinguished by their different lengths. The alleles for each gene are shaded differently in table 4.1. The sex chromosomes are called W and Z, just like we have X and Y. So this one had W and Z. Now, this table is the main thing which you need to really concentrate and look at it very carefully and look at it. Now, female A. And look at what is this here, allele length base pairs. So we have gene locus. Let's look at all very carefully. We have gene locus P, then we have gene locus Q, and then we have R, and then we have S. Now, female A and female B, we were talking of these two were the parents. And then male C was also a parent. So this was WZ, this was WZ, and this was ZZ, the male. So the males are ZZ and the women are WZ, just like we have XY and XX, XY in the male and XX in the female. And then let's look at the gene locus. Now, here when they give you this thing, now this is the allele base sequences like a base pair. So this is 211 base pairs, this is 216 base pairs. So that means this is heterozygous. Now this one again is heterozygous because it's 151 and 154. Then this again is heterozygous, and this is not heterozygous, this is homozygous. Then this is again homozygous. Then this is again homozygous. Then this is again homozygous. And then this is again homozygous. Then let's look at offspring 2 from female A. Now homozygous, 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 and homozygous. But please look at these and these, they are different. This is different again. So 1 and 2 have some differences. They can't be clones, because that's the further question that's coming in the paper. Then female B was again, we had this was heterozygous. Then this is again homozygous because they're both 154. And this is 190 again homozygous. And then this is the heterozygous one. Why? Because the base lengths are different. So that means the proteins are different. The size of the polypeptide chain is different. Then again, we have the different ones. So this is the same. This is now 3. This is same. This is same. No, oh, sorry, I'm using the wrong color. So I'm using the green for homozygous. So this is homozygous, this is again homozygous, this is again homozygous, and this is again homozygous. So you see these two are the ones which are all homozygous. And then we have offspring 4. And in offspring 4 we have again, this is homozygous, 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 homozygous. And there's a little difference between them. You see 3 and 4 were born to be. So 3 and 4 were born to be, and 1 and 2 were from female A. So please do this with yourself. Ask these questions and do this with yourself. Now in male C, what did we have? In male C we had, now this is again hetero, 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 and this is of course then homozygous. 141, 141 were the same. Then we have the last one we talk about was the offspring 5 from B and C. So this is again different now. So how are they going to be if we have them all? This is same and this is same. And then we have the different ones which we are coloring in red. Right? So we've got a different all sort of, so this one is all the same. And then we have, which one is all the same? The rest one. Let's look at the one which is all the same. Uh, this is all the same. This is all the same. Now look at the first part. The first part is using table 4.1. I define gene locus that shows the most genetic variability and name the process that gives rise to different lengths of the allele. So you can see here in R, this is the one which is R. There is the biggest variability, 180, 200, same. So this is different. Let's look at the ones which are different. And this is, okay, we need to finish this video. This will continue on to the next video. Thank you very much.